Hello, this is Fredrik Wormling. I have a research group at the Karolinska Institute in Stockholm, Sweden. We study the immune system with a focus on autoimmunity and cancer. And we use a fair amount of molecular biology tools such as CRISPR to try to understand biological processes, but also finding drug targets. And today I'll go through a script for you, our script, an approach that generates data like this. And the purpose of this is trying to identify connection between genes in a gene set. And we use it to both expand gene sets when we're designing custom CRISPR screen libraries, but also to analyze CRISPR screen, trying to get sort of an idea about, okay, what's the connection between these genes? Are there any functional families, etc.? Yes. Going through the README file here. So to generate this, so this is an R script then. So you need R. I use and we use R Studio also when we're using this, but that's up to you, of course. The script looks something like this, and again there's a README file linked to it. What you need to do, besides having then R and potential R Studio if you want, is to pay attention to these parameters. And that is to define in the script where your reference list is identified. And since this script is based on the DepMap dataset, you need to download the gene effect data from DepMap. That you can do in the, I'm gonna take another one here, sorry, this one here. You want to go for the downloads at the DepMap, so depmap.org, and then go to downloads. It's the CRISPR screen data you want to go for, and then it's the way it looks now, which is February 2015, and there's a download file here of the most current version. Note that there are different versions of their data sets, so I would, although the name of the file you download is not indicating that it's 24Q4, I would call it that just for your own sake. Yes. So you download that, you put it in a folder where you want it to be. The second part is to generate a gene list, and these are the genes that you want to correlate to the DepMap dataset to try to find connections based on their readouts. I'm going to remove myself here. And such list could then look something like this. This is a gene list which is a DNA damage gene set. It's with a particular Go term here. But essentially it's just a text file with a list of genes. You can write them yourself, you can copy them from your RNA-seq experiment, you can take a Go term, you can do whatever you want, any list here. Yes. And then you download that, or you generate that, sorry, and you put it in a folder, preferably the same folder as the reference list. Then you decide which mode you want to go for. So there's a design and an analysis versions you can go for. The design one is going to correlate your list of genes towards all the genes in the DepMap dataset, which means that you're going to get potentially a pretty large data set, right? And this is a way to identify potential genes that could be interested. If you're interested in your gene list, this will identify other genes that are sort of connected to those genes. So if you want to expand your list of genes that, for example, could be part of a custom CRISPR screen, that could be useful. If you use the analysis function, it's only going to correlate genes that were found in your list that you provided. So this becomes more relevant when you perform your CRISPR screen and you want to try to get that sort of that first stage when you're trying to sort of wrap your head around, okay, what are these genes? Are there any connections between them? Are there any pathways that seems to be? And there, of course, you can use different tools to identify if the gene set is enriched in different Go terms or other pathways or gene sets. But here it's based on a DepMap data set, right? And you also want to define the cutoff here, and this is sort of how strong the correlations are. I would advise to start with 0.5, and you could go down to 0.3 or something like that, but 0.5 is a good starting point That is a reasonably strong correlation, I should say. But if you take a too low number here, especially in the design mode, you can start getting really big data sets, and especially if your gene list is long. So the longer your gene list is, if you're using design mode, you have a risk of going for very large files. So then you want to have a higher correlation cutoff, probably. Yes, so those things you need to define. And just a little bit about the data sets from the depth map then. So you know the depth map data set is has a lot of different things. It's a fantastic tool. And one of the things are these collection of full genome CRISPR screens. And here you can find I should emphasize that the screen here is doing something that depth map also can do, although the script is doing it in a bulk mode, so you can do it with a list of genes, while depth map only the portal as it is now at least only allows you to do it one gene by gene. But the correlations we're talking about is something like what you're seeing here. So each dot here is a cell, and on the x-axis here you see how the cells behave when you knock out or you target TP53, and here how they behave when you target MDM2. And this is based on a sort of dropout setting where they take an early time point and a late time point in all of these type of screens. And if it's a positive number, it means that the cells have sort of expanded versus what you would expect, comparing the late time point to the early time point. So the cells like when you remove this gene is keeping them back somehow, so they're expanding more and are more abundant than you would expect at the later time point compared to the early. And if it's a negative number, it means that the cells don't like when you knock it out. You see fewer cells, and of course the larger the number is, the more negatively impacted. And the correlation we're talking about is related to the correlation line here. And the numbers this correlation in the script are using is actually going to be exactly the Pearson, the same number as here. And that makes sense because these two things are based on the same data set, right? Although the calculations in the script are not the same calculations as here, but they generate the same data. So that's the basis for this. We're trying to find connection between genes, how they're behaving, right? Of course, here one should pay attention or sort of note that the correlations are based on survival or proliferation based on an early and a late time point, right? So if you're looking at signaling pathways and the capacity of a signaling that doesn't have anything to do with sort of survival, you might not identify correlations, or so I should say the correlations you identify might not be necessarily relevant for your screen. So these are more lean towards this sort of survival type of phenotypes or proliferation phenotypes. Anyway, so we define all these sort of things, and what we then do is to copy the script. You can of course do this in more elegant ways by saving it as an R file, etc. But this is a simple way to do it. So I just paste it in here. If we want to make any changes here, we can do it, but right now let's not. And then we press enter. These calculations, and again I could just reiterate that we have a gene list now of roughly 200 genes that are linked to DNA damage. Now the output came out here then. And what it's going to do is make a folder in the same folder where your gene list was found. So the gene list is found here. This is the reference file. I put them in the same folder, and then they're going to make a folder. The script makes a folder in the same folder with the date, and then defining if it's design or analysis. And if there is already, if you run it before and there's already a design folder there, it's going to call it 1, 2, 3, etc. So what do you find in here? So first of all, we have a settings and a genes not find here. So this is sort of telling you when did you do it, how did you do it, which settings did you use. It defines which genes were in your list, and also importantly define which genes that were not found. And I'm not going to spend too much time on that, but if this list becomes large, you should try to find synonyms or homologues for these, or orthologs for that matter, that could be found in the data set, in the reference list, as of course you don't, if they're not found, they're not going to be part of your analysis, right? And they could be really important for identifying these sort of connections. So I advise you to look at these, trying to find synonyms, add them to your, in an iterative fashion, sort of going in, adding the synonyms to your list, and then doing it again, right? But we can talk more about that some other time. Yes, so that's sort of a settings file. You get a as I showed you, you get this type of graphical output which shows sort of genes that are correlating. The larger the lines are, the edges, the stronger the correlation. Positive correlations are blue lines, and positive correlation means that the cells are affected in a similar manner if you target these genes. Red line means that they are a negative correlation, which means that they're opposite in how they are sort of affected when you target the gene. And then you start to finding these sort of clusters indicating that, okay, these genes actually could be sort of related to in some way. And these stars here then identified if they are finding your gene list. So here we had AF2AK4, for example, and the script identified that these two are strongly correlated to this one. So if you're interested to target this one, maybe you could target those two also. Yes, you get both a PNG and an EPS file, depending on how you want to work on it. You can change this to a TIFF if you want in the script, but for now it's a PNG and an EPS. You get a clusters output, so this is a comma-separated text file. So you need, if you open it like this, you need to, it makes more sense to separate it. And what you then see is the list of genes. So these are all the genes that were part of the here, but it tells it to you in a different way. And again, the arrows, or sorry, the stars are identifying that they are were found in your gene list, and the other ones here are the ones that the software, or sorry, not the software, the script is identifying to be linked by correlation. And it also defines them. So this is cluster 1, this is cluster 2, etc. And the clusters are arbitrarily named, so that doesn't say anything more than that these genes were part of the same clusters. And in this case, it's this cluster. Yes. So in design mode, if you want to design a custom CRISPR screen target, you could then, you started with one list, and now what you can do is just to sort of copy this whole list, and then you base your screen on those genes. Not the purpose of this video, but of course we do these sort of things, design these sort of screens with green listed software where you can sort of paste the genes and then sort of design your CRISPR screen, custom libraries like this. But again, not the purpose of this video. So, but that's sort of the approach I would do. I think it's pretty useful here. This is the mean and standard deviation based on all the genes then. Sorry, all the cells, data from all the cells, how these cells are affected by targeting these genes. Right? So in the DepMap dataset, it's roughly 1,000 genes, 1,000 plus. Maybe even 1,200. And we talked about it, negative means that the cells don't like when you knock it out. So DepMap defines -1 as a pretty strong effect on the cells, as a common essential gene. So essentially, if you knock it out, cells are not going to be happy, right? So the more negative this number is, more likely is that you just knock it out and the cells are not going to stay, right? There are some genes that have numbers down to sort of -4, and those genes, just you knock them out and the cells are dead, right? So you could consider, in this case, none of these are extreme, these values, so in this case I would probably just take this list and use them for my screen, the design of my screen. But if some of these are really negative, you could consider, do you really need to have them there, because you're going to knock them out and the cells are going to die anyway, and then you don't I mean, depending what you look at, but likely your screen is, you're not going to learn anything from including those genes in your screen. I would argue, at least. So this number could be pretty interesting to look at. And then finally, there is a correlation file. Again, data, text to columns, eliminate, next, comma, chunk. Which essentially is just looking at the correlation between these genes and defining the correlation number. And it also tells you which clusters they are part of, and also if the first or the second genes were part of the list that you uploaded. So I would argue that for if you want to make some sort of publication, I think the EPS file could be good for, and potentially PNG. You likely want a TIFF for publication purposes, but at least they could be useful as outputs. And the clusters file, I think those three could be very useful for you, while the settings and correlations are more sort of reference files for you to keep to sort of understand what's going on. Okay, good. So with that, I've shown you a software that could be used to identify links between genes. Now I use it in the design mode. I'm not going to do it, but if you used analysis mode, you can get images looking like this. So here all the genes were paired. So this is actual data from an actual CRISPR screen. And when you are at that early stage of getting the gene list, it could be very useful to sort of start generating these sort of things, trying to understand, okay, these genes might be connected. There's some sort of families here and here, maybe, to try to sort of wrap your head around and help you generate hypotheses. But here, of course, you don't have the stars, because these are all genes that were found in your list, basically. So these are the two ways we would work with these, either to expand gene sets to identify genes that could, from a rational point of view, be linked based on a depth map way of looking at things, at least, or to try to understand connections in the screen setting. Having said that, of course, this doesn't have to do anything with CRISPR. I mean, the depth map data is based on CRISPR screens, but of course you could just have a gene list that you're interested in for different reasons, like from an RNA-seq data or a proteomics data or so, where you want to see, okay, maybe there is something here. Yes, so with that, I thanking you for watching this far, and I hope you can find this script useful. Thanks.